Hello, my name is Rebecca Krisher, and I'm the Research Director at the Colorado Center for Reproductive Medicine. In this video, I'm going to discuss some of the cutting-edge technologies emerging from basic research laboratories that have the potential to change the way we practice ART. At the end of this video, you should be able to describe key emerging cellular and molecular technologies that have the potential to dramatically impact the practice of assisted reproduction and discuss ethical considerations affecting implementation of these technologies. Mitochondrial dysfunction is implicated in disease as well as in fertility. Mitochondrial replacement therapy, or MRT, has been proposed as a method to avert transmission of mitochondrial DNA defects to children. Several methods have been developed in oocytes or zygotes, such as pronuclear, or PNT, and spindle, ST, transfer. Pronuclear transfer has the potential of higher levels of mitochondrial DNA carryover and suffers from ethical issues regarding donor embryo destruction. Spindle transfer has led to normal development in adults and low mitochondrial DNA carryover in primates. MRT could also offer replacement of deficient mitochondria in oocytes from older patients. The schematic shown here is a representation of both spindle transfer into oocytes and pronuclear transfer with zygote stage embryos. Spindle transfer involves isolation and transfer of the spindle chromosomal complex from an unfertilized patient oocyte containing mutated mitochondrial DNA to the cytoplasm of a donor enucleated oocyte containing healthy mitochondrial DNA. The reconstructed oocyte is then fertilized and transplanted. In pronuclear transfer, this takes place after fertilization, at the one-cell embryo stage containing a male and female pronuclei or zygote. Both pronuclei are isolated and transplanted into a cytoplasm of an enucleated donor zygote. There are lingering concerns about the fate of mitochondria containing mutated DNA that are transferred to the reconstructed embryo during either procedure, although studies suggest the probability of significant carryover is low. There are also safety concerns that children born as a result of this technique may have abnormalities due to unmatched nuclear and mitochondrial DNA. To date, long-term studies in primates have not detected any health issues. At this time, it appears governments may permit this technique to be developed for a specific and defined group of patients whose offspring may have severe or lethal mitochondrial disease and who have no other option of having their own genetic child. Another consideration is that spindle transfer and pronuclear transfer do induce permanent changes to mitochondrial DNA that would be transmitted to the next generation, thus qualifying as germline gene therapy. Finally, children born after mitochondrial replacement therapy would have a genetic connection to 3 individuals, mother, father, and the mitochondrial DNA donor, and could thus be considered as having 3 parents, although the mitochondrial DNA contribution is small. This slide focuses on the production of oocytes from pluripotent stem cells, although sperm production has also been reported. The schematic shown here demonstrates how this is accomplished in vitro, starting with the differentiation of embryonic stem cells into primordial germ cell-like cells. These cells are then mixed with somatic cells to form a reconstructed ovary. After culture and differentiation in vitro, mature oocytes are formed. Bright field images show the progress of reconstructed ovarian growth over a period of 3 weeks. These oocytes can be fertilized and successfully develop to the blastocyst stage and produce pups after transfer. Editing of the DNA of a human embryo to prevent a disease in a baby could someday be ethically allowable. The technologies to achieve this goal have recently been developed and demonstrated in human embryos, although additional research on efficacy, risks, and benefits must first be undertaken. The figure shown here is from the first study in the U.S. using CRISPR gene editing technology to alter the genome of a human embryo. In this study, a mutated gene carried by the sperm was targeted by injection of CRISPR-Cas9 into human zygotes at the S phase of the cell cycle. Embryos were then analyzed at the 4 to 8 cell stage. Injection during S phase resulted in mosaic embryos consisting of non-targeted mutant, targeted non-homologous end-joined repaired, and targeted homology-directed repeat repaired blastomeres. The efficiency of the procedure is shown in the pie graphs, where you can see that the prevalence of embryos with the gene mutation has been reduced. Previously, it has been difficult to study the differentiation of cell types and their organization in the human embryo because to remain viable, blastocyst stage embryos must be transferred to the maternal environment. Shortly after transfer, the embryo implants and becomes enveloped in maternal tissue, further hiding it from study. Recently, establishment of an in vitro system that is able to maintain viable embryos in culture past the blastocyst stage has been reported. This would allow study of peri-implantation human embryo development, morphogenesis, and implantation. The first panel shows representative bright field images of human blastocysts developing in vitro until day 12 to 13. Scale bars are 100 microns. The next panel of images are confocal Z sections of a day 10 to 11 human embryo, stained for OCT4, which detects epiblast cells, APKC, which detects trophectoderm cells, and F-actin, which defines the cell boundaries. The arrow indicates the presence of a small lumen surrounded by radially organized OCT4 expressing epiblast cells. The scale bar here is 50 microns. Boxes indicate the magnified area, scale bar 20 microns. Finally, the last panel is a stylized model of human embryo peri-implantation morphogenesis. The main remodeling events that take place during this transition are segregation of epiblast and hypoblast progenitor cells on day 7, polarization and proamniotic cavity formation in the epiblast on day 8 to 10, differentiation of the trophectoderm into cytotrophoblast and syncytiotrophoblast cells on day 8 to 10, and formation of the prospective amniotic epithelium, the prospective yolk sac, and the bilaminar disc on day 10 to 11. Thanks for watching. I hope this introduction to emerging technologies in ART has stimulated thought about how evolving scientific knowledge may translate into novel treatment modalities in ART and dramatically change the landscape of treatments available for patients.